Hi everyone, am I audible? Audible? Yes. Hi kids, am I audible to you all? Hello. Hi everyone, a very good evening to all of you. Hi kids, I hope I'm perfectly audible and visible to you all. Hello. Can you see me? Can you hear me? Hi Shridhana, Shruti, Avinash, Avni. Hello. A very good evening to all of you. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and it is our lecture 5th on the chapter cell, and today we are going to finish this particular chapter, right? So are you guys excited? We are done with the one chapter of your NEET 2024. Yes. So first of all, let me know in the chat section whether I'm audible or not. Quick. Good evening. So see, we are done with your one chapter. Today we'll finish the chapter cell, and then we will start cell cycle and cell division. Meanwhile, you should do the practice of the question. You can practice more than 50 questions on this chapter, that is cell. It's a very easy chapter, but still sometimes you people get confused. Okay, so you should practice the question from this chapter as well. So tell me about yourself. So how's your classes going? Anything, any confusion that you people want me to answer? Something like that? Yes, everyone? And please participate in the chat. Then we'll start the lecture. Anything that you people want to ask, any confusion, any query related to your syllabus, related to the books, related to new NCERT, related to old NCERT, anything that you people want to ask, and anything related to Avengers batch. Yashika, I can, but I will provide the questions on Unacademy platform, right? I hope you all are aware that from 25th of May, we are going to start our Avengers batch. Are you aware of it? On 25th of May, we will be having our first class of Avenger batch. Right, and it is one of the best batches for the NEET preparation. And let me tell you, bache, if you join this batch, your syllabus will be finished in 6 months, and we will practice the questions. The mock test will be there, chapter wise mock test, full syllabus mock test, everything will be provided to you. And the most beautiful thing is, now this batch is at a price of 14999, but you have some special offer till 26th of May, like till 12 a.m. of 26th of May, you guys are having one offer, right? So by applying that coupon codes, you people are going to get this batch in 9599 rupees. So this is the last chance for your enrollment, because from 25th of May, no matter what, we are going to start the classes, right? We are not going to delay your batch. So on 25th of May, you people will have your first class. Okay, you people will have your first class. So join this batch as soon as possible, and if you want to be the part of this batch, what you have to do, see, open up the description box, you have to click the batch link, right? Then add your number, you can even continue with the email. Then after that, see, Avenger coupon code is already applied, and this unlock 20, it is valid till 26th May only. Okay, till 26th May, you can avail this offer, and you will get this course in 9599 rupees. So in 9599 rupees, we are going to complete your syllabus of class 11th as well, class 12th as well. This fees is for the entire year, not just for 1 month. Okay, and along with that, even the practice test papers will be provided to you, revision classes will be provided to you till your NEET examination. So guys, be the part of this batch as soon as possible. Now let's start the topic, and obviously we are discussing about the cell, a very basic topic, but it is the first chapter that we have started for NEET 2024. So I want you people to understand the basics, right? Right now, do not just focus that, ma'am, finish the chapter in 2 lectures, finish the chapter in 3 lectures. No, it should not be like that. Right now we have the time, so we should utilize that time wisely. So right now, focus on your content. Till December we have to finish the syllabus, and then we need to revise. Okay, so we have time, but you need to be consistent. Okay, so let's discuss what we have covered so far. We started with the cell, right? In lecture number 1, the setup was not proper, so I was not able to discuss anything. So basically you can say that from lecture 2, we started our chapter properly, right, bache? So we have completed the cell theory, the basics of prokaryotic cell and eukaryotic cell, right? The animal cell and the plant cell, that was your homework. You need to check the difference in these two type of cells, right, bache? And then we have covered the cell wall, the cell membrane, right, the endomembrane system. So these are the things that we have covered, right, bache? Prokaryotic cell we have discussed in detail. So today we are going to start from the mitochondria, which is the powerhouse of the cell. What are we going to start today? We are going to start with the mitochondria. And what is mitochondria? The powerhouse of cell. Okay, bache? The powerhouse of cell. This is mitochondria, and this is what we are going to discuss. Okay, but before that, see, do you know about endosymbiosis theory? Anyone? Do you know about endosymbiosis theory? Now don't be like, ma'am, it is not given in NCERT, why are you discussing it? Let me tell you, it is something very interesting. You should listen to me very carefully. It is literally very interesting. So what are we going to discuss now? Endosymbiosis theory. It is related to that mitochondria part. See, when you talk about mitochondria, plastids or the nucleus, right? When you talk about these three organelles, these three are double membrane bound organelles. What are they, kids? They are double membrane bound organelles, right? And out of that, when we talk about mitochondria and plastid, particularly the chloroplast, right, the chloroplast, so we relate it with the endosymbiosis theory. Now you must be thinking that, ma'am, what is this endosymbiosis theory, right? It's a very simple kind of theory. I'm not going to provide you the details of this theory, just the overview. So initially we used to have prokaryotic bacteria, right? Initially we used to have prokaryotic bacteria. You know that. What is a prokaryotic bacteria? The primitive bacteria, right? The bacteria having circular DNA, but it is not having the organelles. The genetic material is not present within the nucleus, right? It's a primitive type, isn't it? It's a primitive type. So what happened initially? Let's say there was one anaerobic bacteria. One anaerobic bacteria. Bacteria is the perfect example of the prokaryotic cell. Whenever we want to discuss the prokaryotic cell, we always start with the anaerobic bacteria. So what am I saying? That let's say there was one anaerobic bacteria and one more bacteria was there, and it was very small sized, and it was an aerobic bacteria. One is anaerobic, means it cannot utilize oxygen. It doesn't depend upon oxygen for its processes, right? And when you talk about this particular bacteria, this aerobic bacteria, right, obviously aerobic means it can use the oxygen. It can use the oxygen. This is the first thing. Now this bacteria was like that, my size is big, right? But still I'm not able to utilize the oxygen, and this bacteria, this is small in size, this is aerobic, it can utilize the oxygen. So this bacteria was like that, I should engulf this bacteria. This large sized bacteria was like that, I should engulf the small sized bacteria, right? They discussed to each other, and they were like, this anaerobic bacteria was like, see, I'll keep you inside me. I'm not going to destroy you. I'm not going to disturb you. I just want one property, that you can use the oxygen, and I want that property in me, right? So what am I going to do? I'm going to engulf you. You can live within my body, right? I will just use one property of yours. Rest you can utilize all the cytoplasm, all the space, do whatever you want to do, but be in me, right? So it is basically endosymbiosis theory. See, the word symbiosis, like mutualism. Two things are dependent on each other, isn't it? This is the meaning of symbiosis, right? Two things are benefiting each other. They are dependent on each other, right? Now the word is endo. Endo means inside. So this large size bacteria is going to engulf this small size bacteria. Now what will be the scenario? You can see here like this. You know, different teachers, different books explain it in a different way, but basically this is the gist of this topic. This is basically the endosymbiosis theory. So now if you look at this, if you look at this particular cell, now in this cell there is one more structure which can even utilize the oxygen, right? So that is how we relate the origin of mitochondria and the plastid. Actually, when you talk about the mitochondria and the plastid, right, bache, as I said, they are the double membrane bound organelles, right? And especially when you talk about the mitochondria, right, what are they? They are double membrane bound organelle. Moreover, these are the organelles having their own circular DNA. These are the organelle having 70S ribosome. So they are more like prokaryotes, isn't it? They are more like prokaryotes, isn't it? So that is why when we talk about the origin of these organelles, we relate it with the endosymbiosis theory. That many years back, you can say that 1.5 billion to 2 billion years back, you know what happened, like not exactly 1.5 to 2.5 billion years ago, but just say there is a prokaryotic bacteria, but that bacteria evolved into a eukaryotic bacteria in that time, right, in that billion years. So this is basically the endosymbiosis theory. Now this bacteria can also use oxygen, right? You know, mitochondria is the powerhouse of the cell. Electron transport chain will be there in mitochondria. So this is how we explain it. So up to this part, all clear? It is not going to come in your NEET examination, but yes, it is important, right? Obviously, it is making our topic interesting. Okay, kids? Okay, so up to this part, all clear? So now let's talk about the structure of the mitochondria. So as I said, it is what? It is the powerhouse of the cell, and it is double membrane bound organelle. Just give me a minute. Okay. So now when you talk about the outer membrane and the inner membrane, you can see the difference, right? Outer membrane is continuous, but when you talk about the inner membrane, you will see these finger-like enfoldings. Okay, so the very first thing that you need to note down is that mitochondria is a double membrane bound organelle having the outer membrane and inner membrane. When we talk about the inner membrane is having some finger-like enfoldings, and for that we use the word cristae. For that we use the word cristae. What is the word for that, bache? For that we use the word cristae, and there is a space in between the inner and the outer membrane. What is there? There is a space in between inner and the outer membrane, and that space is known as intermitochondrial space. Okay, intermitochondrial space. Clear, bache? Intermitochondrial space, and the diagram of mitochondria is important. We are going to discuss it in the plant physiology, so it's better if you will understand it right now. Okay, so outer and inner, and the space in between is intermitochondrial space. This is the first point. Now in the outer membrane, you are going to find some pores, right? We used to call it as porins. What is it? It is porins. Yes, everyone, do let me know in the chat section, all of you. Yes, do let me know in the chart section. What is it? It is porins, pore-like structures, right? Pore-like structures are present in this outer membrane. And when you talk about this outer membrane is rich with phospholipids and cholesterols. If we compare the composition of outer and inner membrane, so outer membrane is enriched with the phospholipid and cholesterol. Just a minute. And cholesterol, and the phospholipid choline is present there basically, right? It is enriched with that. Okay, so cholesterol is present. And when you compare the permeability also, bache, the outer membrane is more permeable in comparison to the inner membrane. Okay, outer membrane is more permeable in comparison to the inner membrane. So outer membrane is there, inner membrane is there, right? So inner membrane is having what? Inner membrane is having the cristae. Inner membrane is having the cristae. Done, bache? Bache, cisternae or cristae? What do you want me to explain? Cristae, these are the finger-like enfoldings present in mitochondria. Cisternae is something different. Okay, so now see here you will find some lollipop-like structure. See, we'll talk about it, but before that, see now here this inner mitochondrial membrane is enclosing some material, and for that we use the word matrix. What is the word for that, bache? For that we use the word matrix, right? Matrix. It is the matrix basically. It is neither solid nor liquid. It is just fluid like matrix is present. And now in this matrix, this organelle is having its own circular DNA. Right. Moreover, here in the mitochondria, there will be the enzymes which will help in the protein synthesis. And moreover, here in mitochondria, you will have ribosome. Right. That non-membrane bound organelle. And which ribosome is present there? 70S, just like prokaryotes. So that is why we relate the endosymbiotics theory. Okay. Because this mitochondria is just like, it is just like your prokaryotic cell, na? 70S ribosome is there, circular DNA is there. So that is why we explain the origin of mitochondria in this way, that one cell engulfed the prokaryotic cell, small prokaryotic cell, and with time it evolved into the structure which is known as mitochondria. Is that clear, bache? Is that clear? So this is what you have to remember, right? It is a repeated question of NEET. They used to ask you that which organelles of the eukaryotic cell have their own ribosome. The one is your chloroplast, another is your mitochondria. The one is your chloroplast, another is your mitochondria. So this is what you people need to remember. Okay, bache? Now, just look at this inner membrane. See, moreover, when you talk about the mitochondria, mitochondria is more in animal cell and it is comparatively less, its number is less in plant cell. Okay. If we compare animal cell and plant cell, so it is more in animal cell. Okay, it is more in animal cell. Are you getting my point? One thing. And moreover, see, if a cell is undergoing respiration, if respiration is too much in a cell, obviously we will be having more mitochondria there, and the inner enfoldings, these cristae, the number of cristae will also increase. Okay, the number of cristae will also increase. This is what you need to remember. And mitochondria is also having different shapes, and this is your homework. You people are going to mention it in the comment section. You need to mention the different shapes of mitochondria. Even mitochondria is having different shape, and this is what you need to tell me. Now let's talk about its inner membrane. So inner membrane will also have, the basic thing is same, the phospholipid bilayer. Okay, that basic thing is same, that there will be also be the phospholipid bilayer. But of course, the composition can vary. Okay, kids, it is the composition that will vary. Okay, so now here in this inner mitochondrial membrane, right, at places you will see some proteins, some protein complexes are present there. Right, some protein complexes are present there, kids, and we used to call them as electron carriers. Right, what is present there? Electron carriers. What is it? Electron carriers. Right, bache? Right, Samiksha, the space between outer and inner membrane is intermembranal space. Okay, or perimitochondrial space, whatever you want to call. Okay, so here the basic structure is same. It is also the phospholipid bilayer, but at places you will see some protein complexes. Okay, these are basically the electron carriers. They are going to hold the electrons. They are basically going to even transfer the electron from one electron carrier to another, from one electron carrier to another, right? And this is making electron transport chain or electron transport system. Why do we call mitochondria as the powerhouse of the cell? Why? Because mitochondria can form ATP, right? Why are we saying that? Because mitochondria can form ATP. See, bache, there is one thing, right? One respiration. Let's say aerobic respiration. When the word is aerobic, it means we are going to use the oxygen. So we eat something, right? Right, let's say the main respiratory substrate in our diet is what? It is the carbohydrate, isn't it? The main respiratory substrate in our diet is what? It is the carbohydrate, right? So what is going to happen, bache? And carbohydrate, and which carbohydrate? The glucose. So glucose will be oxidized in the presence of oxygen, of course. Glucose will be oxidized, and what will happen? The energy will be released. Isn't it, bache? The energy will be released. So you know that there are some steps for it. what is going to happen? The oxygen is there. Oxygen is going to do what? Oxygen is going to oxidize the glucose, and what is going to happen? The energy will be released. There are different steps for that. I think you are aware of glycolysis, Krebs cycle. If you know it, very good. Otherwise, we are going to discuss it in plant physiology, right? So if I talk about that respiration, there are different steps. The first step is glycolysis. Then there will be the Krebs cycle. And finally, the things will go to, finally we will be having the electron transport chain. And then finally, there will be the oxidative phosphorylation. Oxidative phosphorylation. So ultimately here in this inner mitochondrial membrane, we will be having some protein complexes, we will be having some different complexes which will help in, right, which will ultimately help in the formation of what? ATP. And mainly ATP will be formed by this structure, right? As I said, some lollipop like structures will be present here in the in these inner enfoldings, and we used to call it as bache oxysome, right, or F0 F1 particle, or ATP synthase. Okay, so what do we have here? We have oxysome, F0 F1 particle, ATP synthase. So where are they present? Where are they present? On these cristae. Okay, oxysome, F0 F1 particle, or you can also call it as ATP synthase. So bache, what exactly it is? Let me show you. So you can consider one more thing here. Let's say basically inner enfolding is also the membrane. So here in this membrane we will be having two things. First of all, just a minute. See, here we have one membrane, right? So in this membrane, this base is basically what? It is F0. This part which is present in the membrane, which is present in that enfolding, which is traversing here, it is F0 particle. What is it? It is F0 particle. It is basically the base. Now here you can see the head region and the stalk, right? Head region and the stalk. This part is base. So the base is known as F0. The head and the stalk is known as F1, right? The head and the stalk is known as F1. Base is known as F0. The head and the stalk is known as F1. So this is what we have. This is basically the oxysome, right? So it is an ATP. Basically this oxysome is going to help in the formation of ATP. It will play a role in the oxidative phosphorylation. Right? Phosphorylation means addition of phosphate. So from ADP it will form ATP. From ADP it is going to form ATP. Are you getting my point? Are you getting my point? So this is how mitochondria helps in the formation of ATP. That's why we used to call it as the powerhouse of the cell. Okay, powerhouse of the cell. So yes, it is the elementary particle, F0 F1 particle. You can also call it as oxysome. You can also call it as ATP synthase. Right, bache? So with the help of, obviously, with the help of, the proton gradient, with the help of H positive gradient, it is going to help in the formation of ATP. It is going to help in the addition of phosphate to this ATP, and it will form the ATP. So this is what you need to take care. So this is the role of this mitochondria. So you can see one more diagram here, right, bache? The cristae, F0 F1 complex, the membranes, the space in between, and moreover, bache, do you know here in this matrix the Krebs cycle will take place? Here in the matrix of this mitochondria, what is going to happen, bache Gokul? The Krebs cycle will take place. What is going to happen there? The Krebs cycle will take place. And here you can see the structure. So see, inner mitochondrial membrane, the inner membrane part, the outer side, and here you can see the proton will pass, and finally the ATP will form. Okay, the ATP will form. So this is about the mitochondria. So in detail we will discuss it in the plant physiology, right? So here you can see mitochondria, and the singular is mitochondrion. The singular is mitochondrion. So when we use the word mitochondria, we are talking about so many mitochondria, right? Plural it is. So here you can see when it is not specifically stained, we cannot see it under the microscope. If we want to see the mitochondria under the microscope, so what we have to do, we need to stain it. So it is your homework. You people are going to tell me in the comment section after the class, which stain will stain mitochondria? Which stain will color mitochondria? That is your homework. Done? Done? Very good, Anthony. Excellent. Excellent. It is Janus green. You are right. Right? It is Janus green, but later on you have to mention it in the comment section. So this is your homework. Okay. So number of mitochondria per cell is variable. Obviously it depends upon the need. It depends upon the metabolism. It depends upon the physiological activity of the cell. Obviously we have covered this thing, right? So when you talk about the shape and size, obviously even it is different. It is variable. You cannot say that there is a fixed shape, fixed size. No, not at all. Like here you can see it is sausage shaped, or it can be cylindrical, and the diameter is 0.2 to 1 micrometer, bache, right? And when it comes to the length, it is 1 to 4.1 micrometer. So usually you do not get questions based on the dimension, but rarely, right, you can get such question in the NEET exam too. So if you can remember it, good, but if you cannot, it's okay, it's fine. Later on near to the NEET examination, we can revise it again and again. Okay, that's not the problem. So each mitochondrion is a, as I said, it is a so this is the diagram from the NCERT. So here you can say, so it is a membrane bound structure. Outer and inner membrane is there, bache, which is dividing its lumen into two aqueous compartment. Okay, into two aqueous compartment. Like here, you know, outer mitochondrial membrane, the inner one. So inner one here is having the matrix, right? And here is just we are having the space, and here you will be having the cytoplasm. Okay, cytoplasm. So inner compartment, as I said, it is having the matrix, the homogeneous substance, bache, and outer membrane forms the continuous limiting boundary of the organelle. Clear, bache? Clear? So enfoldings are what? They are cristae. So singular as crista. Done? Okay. So see, basically these cristae, these are going to increase the surface area, right, bache? So they are having both the membranes, the outer and inner membrane is having different enzymes. It is the inner membrane which will play a role in the ATP synthesis. Remember this point. It is the inner membrane. It is the inner membrane which is going to play a role in the electron transfer. It is having that electron transport chain. It will play a role in that ATP formation, not the outer membrane. When it comes to the outer membrane and inner membrane, even their permeability differs. Outer membrane is more permeable, but inner membrane is selectively permeable. Even the composition of these two membranes, it varies. Outer membrane is having more cholesterol, it is having more phospholipids. Okay, and where are back and back protein located in mitochondria? Malik, not required for the NEET examination, right? It is as per masters, so you don't need it now. Okay, done. So mitochondria, the site of aerobic respiration, I told you that in the matrix there will be the Krebs cycle, right? And in the inner mitochondrial membrane there will be what? Electron transport chain. So rest, it is having the DNA, it is having the RNA, even the ribosome, 70S ribosome is there. So these mitochondria, they can, that is why we used to say that mitochondria is what, bache? It's a semi-autonomous organelle, right? It is semi-autonomous organelle. Semi means half. Like this organelle, it can divide by its own with the help of binary fission, right? So new mitochondria will form from pre-existing mitochondria because they can divide by binary fission. They can divide by binary fission. This is the first point that you need to remember. Okay, what is the second point that you need to remember here is, now here it is having its own DNA and ribosome and RNA. So it is not totally dependent upon nucleus. Okay, it can synthesize its own protein. It is not totally dependent upon the nucleus. So this is what you people need to remember. Okay, this is what you need to remember. Done, bache? Done. So it will divide by a binary fission. As I said, not totally dependent on nucleus for its protein needs. It can synthesize its protein. Not everything it can synthesize, but yes, still it can do that. So that is why it is the semi-autonomous organelle. And same is the case for the plastids. So this is what now we need to discuss, plastids, in which we will talk about the chloroplast, we will talk about the chromoplast, and we will talk about the leucoplast, right? Chloroplast, chromoplast and leucoplast. Chloroplast, chromoplast and leucoplast. This is what we need to discuss. Okay, this is what we need to discuss. So here you people can see, when you talk about the plastids, they are present in the plant cells. They are not present in the animal cell. It is also a difference. Okay, okay, it is also a difference. When you talk about the plastids, they are present in the plant cell, and in the euglenoids they are not present in the animal cell. Now if you know euglenoids, then good. Otherwise, what are euglenoids? They belong to kingdom Protista, right? Here we are talking about the euglena. Remember, a connecting link between plants and animals. Do you remember euglena? It's a connecting link in between the plants and the animals. Euglena is having the mouth as well. It is having the chloroplast as well. Okay, so it's a connecting link. So euglena, the single-celled eukaryotic cell, belongs to kingdom Protista. So kingdom Protista is having the euglenoids. So plant cell and euglenoids will have the plastids. Right, bache? So we can easily observe them under the microscope. Their size is comparatively large, and they have specific pigments which is going to impart color to the different parts of the plant. If you talk about a plant, you know that leaves are green, and if you talk about the fruits are having different color. Let's take the example of tomato. Tomato is red in color. Why? Because of the pigments. So that pigments are present here in these plastids. Okay, that pigments are present here in these plastids. So basically we have, based on the type of pigment, we have three type of plastids. I told you: chloro, chromo and leuco. Chloro, chromo and leuco. And the most common type is what? It is the chloroplast. And firstly we are going to discuss about the chloroplast. Okay, bache, chloroplast. So the word is chloro, chlorophyll, of course. So of course the color is going to be green. Right, the color is going to be green. So this, because of this chloroplast, because of that chlorophyll, right, the plants are having that green color. So again, when you talk about the chloroplast, again it is also having that double membrane bound structure. But here, bache, you are not going to see that inner enfoldings just like mitochondria. Right, Anthony? Here you will not find that cristae. In the chloroplast, obviously we will be having two membranes. One is the outer membrane and another is the inner membrane, but here you will not find that finger-like enfoldings, not in the mature state at least. Okay, and the space between two is again intermembranal space, and you can even call it as inter or periplastidal space. Right, it is periplastidal space. Now, bache, this inner membrane is enclosing the stroma. Here for the matrix of the chloroplast, we use the word stroma, right? For the matrix of the chloroplast, we use the word, yes, bache, we use the word stroma. What is the word here? It is stroma. And again, chloroplast, the plastids, they are also semi-autonomous, right? When you talk about their origin, again you consider the same theory, endosymbiotic theory, right? Here also you consider the same theory, endosymbiotic theory. So Mamta, here also we are having, right, circular DNA. Here also we are having 70S ribosome. Are you getting my point? And some RNAs are also there, of course. Okay, chloroplast. So you know that when you talk about the chloroplast, the one thing that came in our head is chlorophyll. Okay, what is it? It is the chlorophyll. So when chlorophyll is there, obviously we are going to relate it with the photosynthesis, and plants are going to perform the photosynthesis. So that is why plants are having these chloroplast. Okay, so in this way you can relate. So definitely they are absent in the animal cell. Okay, they are absent in the animal cell. And again, because of circular DNA and 70S ribosome, they are semi-autonomous organelle. Okay, they are semi-autonomous organelle. Now, one more thing. Now, see, here in the stroma, you will be having some, membrane bound structures. Can you see it? We will be having some membrane bound structures. What are they? They are membrane bound structure, right? Let's say here we have this membrane bound structure. So bache, it is membrane bound, right? It is membrane bound. Let's say a membrane is covering it, just like that. So that inner space is known as lumen, right? This inner space is known as, yes everyone, let me know in the chat section. This inner space is what? It is lumen. What is it? It is lumen. What is it? It is lumen. Are you getting it? Now, the structure is, the structure is thylakoid. What is the name of this structure? Thylakoid. Okay, thylakoid. It is the part where the light reaction will take place. We will talk about it in detail in plant physiology. But here I will just give you the overview. Okay, so we have the thylakoids. So inner space is lumen. So thylakoid is what? It's a membrane bound sac-like structure, right? It's a pouch-like structure, membrane bound pouch-like structure. Now, bache, what is going to happen? So many thylakoid, they will pile up. Right, they are going to form a stack. Like let's say you are putting one coin and then over it another coin, right? You are making a stack. So the same thing is going to happen here. This is one thylakoid, over it you will be having another. So you can say that near about 40 to 60 thylakoid will pile up like this, right? 40 to 60 thylakoid will pile up like this. Now this complete structure, it is known as granum. Right, what is it? It is known as granum. Granum means singular, grana means plural. Okay, right. So here you guys can see, so many thylakoids, when they will pile up, right, when they are forming a stack, so now we are using the word granum for it. What is the word bache that we are using here? It is granum. What is it? It is granum. So the number of grana in a chloroplast, it can vary, again depend upon the need, right? And if I talk about the shape of the chloroplast, even it is different, right? Related with the plant kingdom, Chlamydomonas having cup shaped chloroplast, some algae they are having ribbon shaped chloroplast. So the shape of chloroplast can vary, but a lumen is the space in between. Something is membrane bound. Just take the example of a bamboo. Outside stem is there, inside it is hollow. That hollow part is lumen basically. Same is the case here. Okay, so here we have the granum. So granum is singular, grana is plural. So there will be so many grana in a chloroplast, right? Now bache, the another thing is, see, one granum is attached to the another granum, right? One granum is attached to the another granum. So for this we use the word stroma thylakoid, right? For this we use the word stroma thylakoid. You can even use the word fret lamellae. Okay, right? The part, this lamella which is connecting different grana. So for that we use the word stroma thylakoid, right? Or fret lamella. Stroma thylakoid or fret lamella. Even intergranum or intergranal thylakoid, right? It is stroma thylakoid, fret lamella, and you can also call it as intergranal thylakoid. Okay, intergranal thylakoid. Are you getting it? Are you getting it? Now bache, where PS2 is present, PS1 is present, that we will discuss in the plant physiology only. But here again I'll give you the overview. So here you people can see, even in the thylakoid membrane you will find some protein complexes, right? Here in the thylakoid membrane you will find some protein complexes, the one which are going to trap the solar energy. They will when solar energy will be trapped, obviously what will happen? The electrons will get excited. They will move from one, right? They will move from one electron carrier to the another, and in this way obviously the energy will be produced, right? We will again see the proton gradient here, and ultimately here also we will talk about the formation of ATP. Here also we will talk about the formation of ATP. So that detailed process is there in the plant physiology, and we are going to discuss it there. Okay, bache, shayad, are you getting it? Right. So where PS1 is present, PS2 is present, that we will discuss in plant physiology. But as of now, you should know this structure. Okay, you people should know this structure. Okay. So now when you talk about this stroma, here in the stroma there will be so many enzymes. It is fully loaded with enzymes. The enzymes which are used for the dark reaction of photosynthesis. Actually photosynthesis will occur in two two steps. First is the light reaction, another is the dark reaction. Light reaction will take place here in thylakoid, right? When you talk about the dark reaction, it will be there in the stroma. So stroma is fully loaded with the enzymes. Okay, it is fully loaded with the enzymes. It will be having so many enzymes for the dark reaction. So in this way, it is going to form, that glucose, that starch. So this is what we need to remember. Okay, so up to this part, if there is any doubt, do let me know. Yes, if there is any doubt, do let me know. Quick. Do you have any doubt? Sure. So chloroplast having what? Having fat soluble. Different pigments are there na. So it is having fat soluble carotenoids, right? Fat soluble carotenoids which will contain carotenes, xanthophyll. So these are the name of the pigments that are going to impart different colors. Okay, that is going to give different colors. And when you talk about the lipids, right, basically it is in chromoplast. Like I just told you about the chloroplast green in color, right? Mainly chlorophyll is there. Another word was chromoplast. See, chromoplast. So chroma word means color. So it is responsible for the different colors. Like fruits are having different colors, and in the case of plant we just don't see the green color. There are other colors also. So this chromoplast is fully loaded with the pigments. Okay, this chromoplast is fully loaded with the pigment. So in the case of chromoplast, you will see some fat soluble carotenoids, keratin is there, xanthophyll is there. Like if I talk about the color of the tomato, so color of the tomato is because of lycopene. You can note it down, right? The color of the tomato is because of the pigment lycopene. So again, it is also present there, right? And when you talk about the leaves, in the case of leaves, the green color, that green color is majorly because of the water soluble pigment, right? Then it is not the fat soluble pigment, it is the water soluble pigment which is imparting that green color. So this is what you need to know. It is important. Okay, it is important, you should know about it. So just write it down quick. Done, bache? Yes? Done? Oh, just a minute. So here petals, I am talking about the petals. Leaves are the green na, they are because of that chlorophyll only. But when you talk about the petals are, the color of petal is because of water soluble pigment. Again, it is also present in the chromoplast. That water soluble pigment is anthocyanin. So bache, it is a previous year question, you should note it down. It's a PYQ, it's a previous year question. What is it? It's a previous year question. So anthocyanin is also present in the chromoplast, but it is not fat soluble, it is water soluble pigment. And these carotenoids, keratin, xanthophyll is responsible for that yellow colors. During autumn we see na yellow foliage, that is because of this xanthophyll. So they are fat soluble pigments. But when you talk about the anthocyanin, what is it? It is the water soluble pigment. Right, it is the water soluble pigment. So petals are having anthocyanin. So ultimately it is the part of chromoplast. Right, now bache, the leucoplast is colorless. These are colorless plastids. Like there are many parts of the plant, basically majorly the roots, they are not exposed to the sunlight. Okay, so they are not exposed to the sunlight. So in their case also we have these leucoplasts, but what is going to happen, bache? These leucoplasts, they do not have the pigment, so they are meant for the storage. And see, proplastid, it's a common structure. We have a common structure, proplastid. It can form any type of plastid. We have discussed three types of plastids, chromoplast, chloroplast and leucoplast. Chromoplast, chloroplast and leucoplast. So it is the proplastid which is going to make these three type of plastids. Okay, it is the proplastid because the basic DNA is same. So obviously it can form. And even bache, the chromoplast, the chloroplast can become chromoplast, even they can even get transformed. Okay, even they can even get transformed. So when you talk about the leucoplast, there are three words, there are three types leucoplast. One is aleuroplast, another is amyloplast, and the last is Elioplast. And this is what you need to remember, given in NCERT, and it is important from examination point of view, bache, right? Important for MCQs. This part is important for MCQ, so you should note it down in your notes. So when it is Eleuroplast, Eleuron word is related to the protein. So this plastid is going to store the protein. Eleuron layer, right? It is the protein. It is the protein. And now amylose, carbohydrate. So it is the plastid which will store carbohydrate. Okay, it is the plastid which will store carbohydrate. Elioplast, it is for the oils and the fats. Done. It is for the oils and the fats. Done, bache. So this is what you need to remember from this part. So here you can see the chloroplast contain chlorophyll, of course, majorly chlorophyll A, which is a universal pigment. It is present in all the photosynthetic plants, chlorophyll A. So along with that, even the chlorophyll B will be there, but majorly chlorophyll A is present, and the carotenoids. So here you can even mention carotenoids are fat soluble, right, bache? They are fat soluble. So here you can see the chromoplasts, fat soluble carotenoid pigments like carotene, xanthophyll, and others are present. They are going to give yellow, orange or red color to the different parts of the plant, right, bache? Leucoplast, as I said, colorless, right? So they will help in storing the nutrients. So amyloplast will store carbohydrate, example is potato. Elioplast will store oils and fats, bache, and just a minute. And Eleuroplast will store the protein. So it is important. Done? It is important. Clear, bache? So majority of chloroplasts are green colored. You know that they are found in the mesophyll cells of the leaves. Mesophyll cells. When we talk about the internal structure of the leaf, internal structure of the leaf. So the cortex in the case of leaves is known as mesophyll. It is basically the mesophyll. So it is fully loaded with the chloroplast for the photosynthesis. Okay, for the photosynthesis. So see, the lens-shaped chloroplast is there, oval, spherical, discoid, or even ribbon-shaped chloroplast is there. So this is your homework. You need to mention the examples. Okay, which plants are having lens-shaped chloroplast, which plant is having cup-shaped chloroplast. This is what you need to tell. And when you talk about the number of chloroplasts, in Chlamydomonas, cup-shaped chloroplast is there, and there is 1 per cell. And in green algae, 20 to 40 chloroplast can be there in per cell. Okay, so mitochondria and chloroplast both are double membrane bound. I told you already. So bas, that's it, that's all. So here you people can see the diagram as well. So I hope each and every word is clear to you. So if still there is any doubt, do let me know. Do let me know. We are done with this thylakoid part. We are done with this this circular DNA, 70S ribosome, everything we have covered. But still, if there is any doubt, do let me know. Quick. Done, bache? Yes, bache, in bundle sheath cells also, we'll talk about Krebs anatomy. We'll discuss everything in plant physiology. Cell chapter is just the basic. So I think if the chapter cell is clear, then you know about the cell, you know about the organelles, you know about the functions. So individually, whenever we will discuss it, then it will become easy for you. Okay. So done? Sure? Fine. So the next is ribosome. Although we have covered it in the prokaryotic cell, but still, let's revise it again. So you know that ribosome is the protein factory. What is ribosome? It's the protein factory, right? It is going to synthesize the protein, and it is what? It's a non-membrane bound structure. Okay, it's a non-membrane bound structure. Clear? It's a non-membrane bound structure, and it is present in both, in the eukaryotes as well, in the prokaryotes. So in eukaryotes, cytoplasm will contain 80S ribosome, bache, and mitochondria and chloroplast will have 70S ribosome. So we can say that in the case of eukaryotic cell, both the ribosomes are there, 80S also, 70S also, 80S also, 70S also. And I have already explained here that S is the Swedberg unit. It is basically the sedimentation coefficient. It is the sedimentation coefficient, bache. Okay, I explained you the cell fractionation in our first lecture. So I have already explained it. You can check the first lecture, right? So George Pellet in 1953, he observed it under the electron microscope as dense particles. He was the one who observed it. Okay, he was the one who observed it. And when you talk about the structure of the ribosome will be having, you know, proteins. Ribosomal proteins are there. It is made up of ribosomal RNA. So ribosomal RNA and ribosomal proteins used to make ribosome. Ribosomal RNA and ribosomal protein used to make ribosome. Is that clear? Ribosomal RNA and ribosomal proteins used to make ribosome. Clear, bache? Clear? So large and the smaller subunit is there. In 80S, the large subunit is 60S, the smaller subunit is 40S. Okay, so they both will join at the time of protein synthesis only. And how? With the help of magnesium ion. Okay, with the help of magnesium ion. And in the case of 70S, the larger subunit is 50S and the smaller subunit is 30S. Again, with the help of magnesium ion at the time of protein synthesis, it will join. And moreover, if you remember, in the prokaryotes to one mRNA, so many ribosomes are attached. Remember, bache, so many ribosomes are attached. So it is known as polysome or polyribosome. It is the polysome or polyribosome. Clear? Clear? So bache, here, even the 60S, the 50S, the 40S, as I said, it is made up of different type of RNA. Like if I talk about the 50S, the one is 23S ribosomal RNA. This 50S itself is made up of different types of ribosomal RNAs and different type of ribosomal proteins. So I am giving you the example of one ribosomal RNA, that is 23S. So this 23S is also a ribozyme. Do you know the meaning of ribozyme? Do you know the meaning of ribozyme? Yes. What is ribozyme? Anyone in the class? What is the meaning of ribozyme? Ribozyme. When RNA is acting as an enzyme, when usually proteins used to make the enzymes. Mostly enzymes are proteins, they are made up of proteins. But bache, when RNA start functioning like an enzyme, exactly bache Anthony, when RNA become catalytic, so at that time we use the word ribozyme. So what is ribozyme? When RNA acts as an enzyme. So when you are talking about this 50S ribosomal, a large subunit of this ribosome, so it is itself made up of different type of ribosomal RNA. So out of that, one is 23S. Right, out of that, one is what? One is 23S. It is 23S. So this 23S ribosomal RNA, which is making this larger subunit, right? It is also the ribozyme. What is the meaning of ribozyme? Catalytic RNA. When RNA is acting like an enzyme. Okay, when RNA is acting like an enzyme. Are you getting it? When RNA is acting like an enzyme. So this is your homework. You need to mention it in your notes as well and even in the comments as well. What is the homework? You need to mention that 60S is made up of which different types of RNA. 40S is also made up of different types of ribosomal RNA. You just need to mention that, and same for 50S and 30S. Okay, so no need to add it. 60 plus 40 is going to be 100, but here this 60S and 40S is showing the sedimentation coefficient. Okay, you know, when I was in class 11th, I used to make this mistake. Here, how is it possible? 50 plus 30 is 80. So it should be the part of larger subunit. It is not like that. The S is indicating the sedimentation coefficient, right? It is related to that weight at which they are going to settle down. So this is what you need to take care. Okay? This is what you people need to take care. So this is about the ribosome. Done, bache? This is about the ribosome. So next is the cilia and flagella, and I have seen that many times students, they don't know exactly the structure of cilia, the structure of flagella, yeah, what is centriole, right? So this is what we are going to discuss today. So today I am thinking that we should finish this chapter, isn't it? See, so less number of students are there. Now you are thinking the cell chapter is very basic, ma'am, you should finish it in one shot or something. Always remember one thing, right? Just remember one thing, one-shot lectures are for those students who are done with the topic, they know the topic well, they know each and everything and they just want to revise the topic. Okay, and when you are starting your revision again, now you are starting your revision again, or you can say that you have started studying again for NEET 2024. So you need to finish your syllabus properly. You should understand each and everything, and even you have sufficient time as of now. If you will start after 2 months or 3 months, then again you have time, but obviously not too much. Right now, if you will do proper planning, if you start studying properly, if you start revising properly, then there will be no problem at all. Till December you people can finish your syllabus. Then in January, February, March, April you can revise again. Okay, and even if you are targeting NEET 2025, then this lecture is for you for sure, because I'm just teaching from basic to the advanced level, right? From basics I'm teaching. Okay, so now we will talk about the cilia and flagella. So what do you know about cilia and flagella? Acha, flagella is motile. Anything else? Class, this session will be for 2 hours. Very easily, I'm going to take that much time. And let's finish it. So tomorrow, what do you have to do? Tomorrow I'm not going to take any live session, right? I'll post one video, the mistakes that you should avoid while preparing for the NEET preparation. Okay, so that video you people should check. So if you're doing that mistake, don't do this, first thing. Okay, and secondly, I'll tell you how to cover the backlogs. So tomorrow I'm not going to take any live session. Okay, so tomorrow you have to revise this chapter cell. You need to practice at least 50 questions from this chapter. 50 questions from this chapter. So day after tomorrow, we will start with the cell cycle and division. Another important chapter, another basic chapter. Done? 9 plus 2 arrangement. Whatever you know about the cilia and flagella, just let me know in the chat section. Quick. No, as of now there is no question practice session. You need to practice it by your own. Done? Okay, so you are right when it comes to the cilia as well the flagella, they are the motile structure which will help in the movement. But before understanding cilia, the internal structure of cilia, before understanding the internal structure of flagella, you people should know about the cytoskeletal elements, right? What you need to know? You need to know about the cytoskeletal element. Okay, so tell me something about the cytoskeletal element. It is not given in NCERT, but most of the time question used to come from this part, like given in NCERT but in a different way. So we need to understand it first. Bache ma'am, in Avengers batch weekly 6 days class or 4-day class will be there. Mahi bache, you will get the schedule, but you can say that per day you will be having 3 or 4 classes. Okay, so in first week, because after I think on 29th or 30th I will not be available. Okay, so I'm going to take your class on Saturday also. After starting the batch, we are going to start the batch from 25th, no? So 25th, 26th, on 25th, on 26th you will be having biology class and one extra class of biology on 27th as well. Then there will be a gap. So we'll share the schedule with you people. Don't worry, you will be having one orientation session. You will get to know about it. Don't worry about that. See, everything is properly planned in the Avengers batch. When you talk about the Avengers batch, we have the number of classes and everything is planned in such a way that you will get time for the self-study as well, right? You are not going to get biology classes daily, not initially at least. After some time, obviously I will increase the frequency of the classes because we need to finish the syllabus. Okay, we need to finish the syllabus. So after some time even I will increase the frequency of the classes because I want to finish it before time and then I want more and more revision in the biology. So classes schedule, the test and everything will be provided to you on 25th of May. Even you people will get one orientation session. So chill. And don't worry about the June schedule, bache. You will get it, Mamta. You will get everything. Whatever we have promised, you are going to get it. Anything else? So can we continue with the cytoskeletal element? See, cyto word is related to the cell, of course. Hai na? Cyto word is related to the cell. Skeletal means skeleton. Like in our case, we have the bony skeleton, right? We have this bony skeleton, isn't it? Skeleton is something which is going to provide the shape to our body, right? It is helping that muscles to get fixed. It is keeping everything intact. You know the role of the skeleton in our body. But when you talk about a cell, in the case of a cell, okay, eukaryotic cell is having a nucleus. It is having the different organelle as well. They are even transporting the things from one organelle to another. So how all that things are possible? How this cell is maintained in a proper shape, right? Of course, the answer is cytoskeletal element. Right? There are some proteinaceous structures which are present in a cell, which are going to provide it a proper shape, which will help in the transport of things, which are even going to generate some energy, which will help in the movement as well. If I talk about the cilia, if I talk about the flagella, right? Cilia, flagella made up of microtubules. But what exactly is that microtubule? The proteinaceous structure, the part of cytoskeletal element. So cytoskeletal element is something which is going to provide support to the cell, which is going to provide shape to the cell, which will help in the transport of the things, in the movement of the, right? Like if I talk about pseudopodia, right? You all are aware of amoeba, isn't it? You know that amoeba is a eukaryotic cell. It's a eukaryotic cell. It's a single, unicellular eukaryotic cell, belongs to kingdom Protista, amoeba. Now in the case of amoeba, we talk about the false feet, isn't it? We talk about the false feet. And what is that false feet? It is pseudopodia. Pseudo means false. Podia word is related to the foot, feet. Pseudopodia. So how that formation of pseudopodia is possible? Again with the help of cytoskeletal element which is present beneath the plasma membrane here, right? If you talk about the cell division, in the cell division we discuss the formation of spindle fibers. We talk about the formation of spindle apparatus, isn't it? So how is it possible? Obviously it is also possible with the cytoskeletal element. So what we need to know now, we need to understand what exactly is this cytoskeletal element. So when you talk about the cytoskeletal element, there are three things that we need to know. Okay, not in detail but still, right? Cytoskeletal element. So we will be talking about microtubules. We'll be talking about microfilaments, also known as your actin filaments, and then we have intermediate filaments. Right, then what do we have? We have intermediate filaments. So the very first thing is microtubules, then microfilaments, then intermediate filaments. Microtubules, microfilaments, intermediate filaments. बच्चे microtubules, tubule, from tubule you need to remember the word tubulin. So microtubule is itself made up of tubulin protein. Ultimately these cytoskeletal elements, they are nothing, they are the proteinaceous structure. What are they? They are the proteinaceous structures. What are they? They are the proteinaceous structure. So microtubule is made up of what? It is made up of tubulin protein. It is made up of what? It is made up of tubulin protein. Now when it comes to the microfilaments, actin, remember actin, in the case of muscles also we discuss actin, myosin, example of microfilament. And then comes the intermediate filament. So basically it is as per the size. It is as per the size. Like when you talk about microtubules, as I said, it is alpha, it is tubulin protein. Actually what is happening, let me show you bache. Let's say this is a microtubule. Actually it is a polymerized structure. It is a polymerized structure. It is a polymerized, like so many alpha beta tubulin, right? Let's say this is one stick, okay? There is one more, and together all that sticks are making one bundle. That is microtubule. Like let's say here, just take this example. This is one stick, this is another, this is another. All of them together they are making one structure. This is microtubule. When you talk about the microtubule, ultimately its monomer is tubulin protein. It is made up of tubulin protein, but there is a kind of arrangement there. Like let's say alpha beta tubulin, alpha beta tubulin, alpha beta tubulin. Many alpha beta tubulin will join. they will form such protofilaments. Right, many alpha beta tubulins will join, they will form such protofilaments. Many protofilaments will join and then they will form one microtubule. Are you getting my example? Right, are you able to understand it? What am I saying? I am saying that ultimately this microtubule is made up of tubulin protein. It is made up of what? It is made up of tubulin protein. We have alpha tubulin, beta tubulin, alpha tubulin, beta tubulin, alpha tubulin, beta tubulin. So many tubulins, alpha beta, alpha beta, alpha beta, alpha beta. They will join, they will form one protofilament. Right, they will form one protofilament. Now so many protofilaments will form together. Together they will form what? They will form this bundle. That bundle is basically what? It is microtubule. So that is how they form. And when you talk about these bundles, right, like let's say, see, it is made up of, this structure is made up of so many filaments. Okay, so their diameter, on the basis of their diameter we are saying that one is microtubule, another is microfilament, another is intermediate filament. So what do you need to remember from this part? That it is made up of tubulin protein, done, alpha beta tubulin. Microfilament, actin filament. It is made up of actin protein, even myosin protein is there. Intermediate filament may you have the examples of desmins, right? Some other proteins are also there. Okay, some other proteins are also there. So remember, microfilaments, they are contractile proteins. They can undergo contraction. Remember in the case of muscles, bache, we talk about the actin myosin. We talk about the actin myosin. Thank you, Shanti. Now focus here, bache. Right? Do you remember in the case of muscle contraction, we discuss actin myosin, actin myosin. So they are proteins. They can undergo contraction. They can contract. Right? They can contract. Right, so microfilaments are contractile proteins. They can undergo contraction. So when you talk about pseudopodia, pseudopodia is formed with the help of these microfilaments. Right, and when you talk about microtubules, your spindle apparatus is made up of these microtubules. Right, cilia flagella, the internal structure of cilia flagella is made up of these microtubules. Moreover, the centriole, the organelle, the membrane bound organelle which is present in animal cell, it is having microtubule. Right, now when you talk about the nucleus, you know the nucleus is also double membrane bound structure. When you people talk about the nucleus, you know the nucleus is also a double membrane bound structure. We have the outer membrane, we have the inner membrane. So in the nucleus, just below the inner membrane, you will find some meshwork of protein. Right, just below this inner membrane, Mamta, it is important, please focus. Just below this inner membrane, you people will find the meshwork. You guys are going to find the meshwork of protein. So it is basically the nuclear lamina. This is the word that we use for it. So it is basically intermediate filament. It is basically the intermediate filament. So that is how different type of cytoskeletal element it helps. Okay, so you people can see here. So microfilament made up of monomers. So many monomers join, they will form it, actin. Like actin, G for globular actin. So many globular actin will join, they will form F actin, fibrous actin. So you can see here. Now see, its diameter is near about 6 nanometer. Now when you are talking about the microtubules, it is near about 25 nanometer. So many protofilaments, they will join and they are going to form one microtubule. So it is all about the diameter. So intermediate, the size is in between microfilament and microtubule. See, 0.10 nanometer. Sorry, 10 nanometer. So coiled dimer. So keratin protein is the example. Desmin is the example. There are different proteins which are the examples of intermediate filament. So here also you can see they are present at different places. Microtubule, actin is present just below the plasma membrane. Intermediate filaments are there which help in attaching one cell to another. So that is how these cytoskeletal elements are playing the role. So I have added few points. I'm not saying that you should remember all the points, but at least you should know the basics. So microtubules, holotubules consist of 13 columns, or you can say that 13 protofilaments of tubulin protein, and in microfilaments 2 intertwined strands of actin like this, right? In intermediate filament, fibrous proteins, supercoiled they are present. Done, bache? So here Keratin family proteins are present here. Tubulin is there. In the microfilament, mainly it is actin. So microtubules help in maintaining cell shape. They help in cell motility, that is cell movement, that is cilia and flagella, right? They help in the chromosome movement and cell division, and even in the organelles movement. So yes, the question can come in exam. They can ask you which cytoskeletal element will help in the movement of the organelles. So the answer is going to be microtubule. Which cytoskeletal element helps in the formation of spindle apparatus? The answer is going to be, what is the answer? The answer is going to be microtubules. Right, bache? So microfilaments will help in maintaining cell shape. It will help in changing cell shape. Helps in muscle contraction. And when it comes to the cytoplasmic streaming, the word is cyclosis. The word is cyclosis, cytoplasmic streaming. So for that, your microfilaments used to play the role. Okay, bache? Microfilaments used to play the role. Okay? And do you remember cell division, animal cell division? The formation of cleavage furrow. That cleavage furrow is also in that there also the microfilament is playing the role. So this is what you people need to remember, right? Done? Okay, so now what we need to understand, few structures. So we are going to start with the centriole. Right, we will talk about the inner structure of the centriole. Okay, so if there is any doubt, do let me know. We'll finish it till 11. Okay, till 11 we'll finish this chapter, but as of now, if there is any doubt, just let me know. Quick. Yes, bache? Sure? Okay. So, bache, centriole is another non-membrane bound organelle. What is centriole? It is another non-membrane bound organelle, and it is present in, okay, it is present in animal cells. It is absent in plant cells, it is just present in animal cells. And as I said, bache, it is a non-membrane bound organelle. It will duplicate in, this is the famous question of NEET examination, it duplicates in S phase. Okay, it duplicates in S phase. Okay, so bache, in the animal cell, these centrioles help in the formation of spindle apparatus. Actually, what is going to happen? Two centrioles. Right, this is important, please listen to me carefully. Two centrioles which are 90 degree to each other, which are perpendicular to each other, they will be placed. These two centrioles are covered with pericentriolar material. These two centrioles, they are covered with pericentriolar material. Can you see this? It is covered with pericentriolar material. And what are they making? They are basically forming the centrosome. The structure that they are making is centrosome. What is it? Centrosome. Bache, there is a difference. Yes, bache, Prashad, you are right. Right, there is a difference in centriole and centrosome. Centriole is a non-membrane bound structure present in animal cell. So centriole itself is made up of microtubules. Right, it is made up of microtubules, and it is having 9 plus O arrangement, 9 plus 0 arrangement. I will explain the internal structure as well. Right, I will explain the internal structure as well. So what type of arrangement is there? 9 plus 0. But sometimes students, they don't even know the difference in centriole and centrosome. So when you talk about the centrosome, when two centrioles perpendicular to each other covered with the pericentriolar material, this structure is basically the centrosome. It is also known as diplosome. It is also known as cell center. It is also known as cell sphere. Okay. So it is centrosome, it is diplosome, cell center, cell sphere. Right? It is centrosome, diplosome, cell center, cell sphere. Okay? Alright. Just a minute. I have a question to ask. May I? So when centrioles are covered with pericentriolar material, it is centrosome, diplosome, cell center, cell sphere. Again, non-membrane bound structure, and it is the one which will help in the spindle apparatus formation. Now, leave it. I want to ask one question. May I? Tell me, may I? Yes, but quickly answer it in the chat section. I'm asking something. I want to ask one question. May I? है ना? मतलब some students are saying, ma'am, RCB lost again, and then they are comparing this platform with some another platform, why it is better, why that is better. So like, what is the point of writing such messages here? मतलब I really want to know that. Whenever anyone spam in my class, I just want to know what is that feeling. Like, why? What do you feel? Euphoria? I want to know that. I literally want to know that. Why do people spam? What is the need? Why do they waste their time? Matlab, are they that lonely? They don't have people to talk? Seriously? Chal, next part. So this is about the I don't get irritated. I'm chill. I'm totally chill. Okay. Very good, Samiksha. I like the answer. Okay. So now what are we going to discuss? We are going to talk about the internal structure of the centriole. What are we going to discuss? We are going to talk about the internal structure of the centriole. Right? Internal structure of the centriole. So, bache, as I mentioned that centriole is having 9+O arrangement. Now, what is the meaning of 9+O arrangement? Ultimately, centriole is made up of what? It is made up of microtubules. It is made up of what? It is made up of microtubules. Right? But how that microtubules are arranged? Bache, always, I'm repeating this part again. Just imagine there is one stick which is itself made up of 13 sticks. Remember it like this. Whenever you talk about the microtubules, just remember, Samiksha, there is one stick which is made up of, Anchal, many sticks. There is one stick which is made up of many sticks, like 13 different sticks. They are joining and they are making one stick, right? Like, so 13 different monomers, they used to make one polymer. In the same way, you can remember it. So basically this is the one stick. So how will it form? With the help of tubulin protein, alpha tubulin, beta tubulin, alpha tubulin, beta tubulin, alpha tubulin, beta tubulin, alpha tubulin, beta tubulin, alpha tubulin, beta tubulin. So that alpha beta is going to form one protofilament. Now are you getting my point? Alpha beta is going to form one protofilament. Okay, so same way we have another protofilament made up of alpha, beta, alpha, beta, alpha, beta. Now that different protofilaments will join and together they are making one microtubule. Okay, together they are making one microtubule. This is what you need to remember. So when it comes to the centriole, we used to say 9+O arrangement. What is the meaning of that 9+O arrangement? Let me tell you. Like, let's say, and remember now, for centriole we also use one word that its structure is a cartwheel like structure. So actually what is happening when you talk about the periphery, when you talk about the boundary, here we will be having three triplets. Okay, A, B and C, right? A, B and C means three microtubule filaments are joined there. We have A, we have B, we have C. What do we have? We have A, we have B, we have C. What do we have? We have A, we have B, we have C. We have A, we have B, we have C. Srinu, 9+2 arrangement is there in cilia and flagella. I am talking about centriole, right? We have A, we have B, we have C. So if you will see, C is incomplete types. Rest A and B is complete, like 1, 2 and 3. Okay? 1, 2, 3. Okay? 1, 2, 3. Like this. Are you getting my point? Right? So on periphery, this is what we have. So A, B and C, the triplet. It is a triplet, isn't it? It's a triplet of fibrils, isn't it? It's a triplet. It's a triplet of fibrils. It is the triplet of these microtubule fibrils, isn't it? A, B and C. A, B and C. So C, A is also made up of these protofilaments, B is, C is. Are you getting my point? 9 plus O arrangement, right? A 9 plus O arrangement we are discussing. A, B and C. Now again, A, B and C. Okay. Just have a look. But just have a look. It is the centriole, right? It is the centriole. In the centriole, right? We used to say the arrangement is 9 plus 0. 9 plus 0 means in the center you will not find any microtubule singlet. You are not going to find, right? You are not going to find any microtubule singlet here, right? Or any microtubule here. But if you talk about the periphery, right? Here on periphery, there are going to be 9 triplets present there. 1, 2, 3, 4, 5, 6, 7, 8, 9. I'm using the word 9 triplets. Triplets means triple, 3, right? 9 triplets are present on periphery. 9 triplets are present on periphery. Mamta, are you getting it? 9 triplets are present on periphery. Means on periphery there will be total 9 microtubule triplets. Why am I using word triplet? A, B and C is present. Now there will be the AC linker. This C will join with the A. This C will join with the A. This C will join with the A. So there will be the C and A linker. Again, here also proteins are going to link one triplet with another, right? The proteins are going to link one triplet with the another. Are you getting it? Are you getting it? Structure is just like that. And here in the center, you will find many proteins. It is fully loaded with proteins. So we used to call it like a cartwheel-like structure. Cartwheel-like structure. Ultimately, it is made up of what? It is made up of what? Microtubules. Complementary to each other. I didn't get it, bache. How come complementary here, Yashika? Please explain it to me. See, this is the structure of centriole. So you can see the triplets here. A, B and C. A, B and C. A, B and C. So C of one microtubule triplet will join with the next A. C will join with the next A. And here also in the center, what will you get, bache? You will get these radial spokes. You will get this central hub. So again it is also made up of protein. So in the center you are not getting any microtubule. That's why in centriole we only used to say 9 plus 0 arrangement is there, or 9 plus O arrangement is there. Right? 9 plus O arrangement is there. So it is cartwheel-like. It is cartwheel-like. Right? What type of structure is there? It is just like a cartwheel. It is just like a cartwheel. The first point is clear or not? Yes, bache. See, in the NCERT, what is given for the cytoskeletal element? An elaborate network of filamentous proteinaceous structure which is having microtubules, microfilament, intermediate filament. So you should know the examples. Okay. So they help in the many functions like mechanical support, motility, and maintenance of the ship. So I have explained it individually, and even I have explained you the structure of the centriole, right? See, the centrosome and centriole is given in NCERT. So centrosome is an organelle usually containing two cylindrical structures called centriole, right? So these are covered with the amorphous pericentriolar material. Actually, it is the pericentriolar material which will help in the spindle fiber formation. It is the pericentriolar material which will help in the polymerization of that tubulin protein, right? The centriole is going to keep it in a proper shape, but actually that polymerization is done by the surrounding pericentriolar material. That is why in the plant cell, even they do not have the centriole, still in plant cell, spindle apparatus used to form. You know, in the plant cell, they do not have the centriole, but still in their case, there will be the formation of spindle apparatus, right? But that spindle apparatus will not be proper because centriole is absent there. This is what you need to remember. So both the centriole in a centrosome, they lie perpendicular to each other. It has an organization like the cartwheel. So I hope it is clear to you now, right, bache? So they are made up of 9 evenly spaced peripheral fibrils of tubulin protein. So now this line is clear to you. What is the meaning of this line? 9 evenly spaced peripheral fibrils of tubulin protein, and each of the peripheral fibril is a triplet A, B and C, right? So adjacent triplets are also linked. A and C linker is there, A and C linker is there, right? So central part of the region is proteinaceous and we used to call it as hub. So hub is basically made up of what? It is made up of proteins. Kankya, ma'am, what is that? But a pericentriolar material is also something like in the mitochondria we talk about the matrix having different composition. Same in the case of centriole, we are also having some substance which is covering this centriole, and for that we are using the word pericentriolar material. Right now I cannot comment over the exact composition, but that pericentriolar material, right, again obviously that basic things are present there which will help in the nucleation or polymerization of microtubules. Okay, so hub is connected with the tubules of the periphery by radial spokes. So even in NCERT they didn't give the structure, but they have explained it. So see, these are the spokes which are connecting this hub to the peripheral fibrils, right? So it is given in NCERT, it is not extra, it is given in NCERT. Done, bache? So here you can see the centrioles form the basal body of cilia or flagella and spindle fibers that give rise to the spindle apparatus during cell division in animal cell. So what the centriole is making? What was the need to explain you the centriole? So actually what is happening, bache? This centriole is also making the basal body. So centriole is having an arrangement of 9+0. This centriole is making the basal body. Now you must be thinking that, ma'am, what is the basal body? Right, basal body is a structure from where your cilia or flagella will arise. Right, see, from this basal body your flagella will arise or your cilia will arise. So at the base of cilia or even at the base of flagella you will get this basal body. So who is making that basal body? Who's making that basal body? The centriole. So can I not say that ultimately this basal body is also having the arrangement of 9+0? Can we not say that this basal body is also having the arrangement of 9+0? Yes or no? We are talking about the eukaryotic cell here, not the prokaryotic cell. Ultimately your centriole is making the basal body, so obviously in the centriole 9+0 arrangement is there. So even in the basal body the arrangement is 9+0. But Samiksha, it is making what? It is making flagella, but that flagella will be having 9+2 arrangement. Even if that basal body is making cilia, cilia is also having 9+2 arrangement. Okay, so cilia and flagella both are, right? Cilia and flagella both are helping in the movement. They are the motile structure. Cilia small in size, bristle-like. Flagella is long, right? Long. The movement of cilia and flagella also varies, Okay, the flagella is having undulating movement, okay, are you getting it? बच्चे याशिका अल्फा बीटा ट्यूबुलिन का ज्यादा नहीं याद रखना है, बस अल्फा बीटा these are the kind of monomers which are together making one protofilament of microtubules. That's all बच्चे. In the eukaryotic cell your flagella is made up of microtubules, right? In the eukaryotic cell your flagella is made up of microtubules, but when you people talk about the prokaryotic cell, when you talk about the primitive cell, in the prokaryotic cell the flagella is made up of flagellin protein, then it is not made up of your microtubules, then it is made up of your flagellin protein. This is what you need to remember. Hub is the central part, pranagya, which is made up of protein. Simple it is. Okay, it is made up of protein, the central part. Done. So now we have to discuss the structure of this flagella. Now let's say this is flagella. Here in the case of flagella, just have a look. See, this is a flagella. Okay, it is a flagella. So this flagella is having outer covering. We are calling it as plasma membrane. This outer covering is basically the continuation of plasma membrane. So it is the plasma membrane. Now here within this plasma membrane, this part, this rod-like structure, right? It is the axoneme. It is the axoneme, right? It is the axoneme. Like we have the plasma membrane. Within this plasma membrane, the rod-like structure is there. For that rod-like structure, we are using the word axoneme. And here within the plasma, this matrix is present, and in between this central rod-like structure, it is together making the structure of flagella. And this rod-like structure is nothing. It is that 9+2 arrangement of microtubules. Okay. It is nothing but that 9+2 arrangement of microtubules. What is it? It is that 9+2 arrangement of microtubules. Now we have to understand the internal structure of cilia and flagella. Then you know that we are left with the nucleus part. Then the chapter will be over, and the microbodies are pending. Then the chapter will be over. So you can see this chapter is so easy, right? So let's finish it today. Done, bache. Keshav, I'm very happy today. Very good, bache. You should stay happy. That's good. That's nice. Very nice. Okay. Now 9+2 arrangement. What is the meaning of 9+2 arrangement? Now here again, nine peripheral, two central microtubules are there actually. In the cilia and flagella, 9 peripheral and 2 central microtubules are there. 9 peripheral fibrils, peripheral, and even they are doublet. Doublet means double they are. 2 central, it is present here, right? And it is singlet. It is singlet. But focus on each and every word. You people should make the notes. Okay, you people should make the notes. 9 is peripheral doublet, 2 is central singlet. 9 is peripheral doublet, 2 is central singlet. Okay, see, in the structure of centriole, we were saying that it was peripheral but it was triplet. It was peripheral but it was triplet. Three fibrils, triplet fibrils were there. Now when you are comparing it with the cilia or flagella structure, peripheral, 9 peripheral, they are what? They are doublet, A and B. B is having this arm. Again, A and B. Then these arms are present. Okay. Can you see it? Now can you see it? It is the doublet. 9 peripheral doublet and 2 central. Means in the center we are having 2 microtubules, but they are singlet, single 2, and they are connected together by this bache, by this central bridge. They are connected together by this central bridge, and moreover these central singlets are covered with the central sheath also, right? They are covered with the central sheath. Now these central microtubules, they are connected to the periphery, and again here we will be having what? We will be having the radial spokes, right? They are connected here, but they are connected with the radial spokes. So now that is why, Anuradha bache, here we use the word 9+2 arrangement. Here we use the word 9+2 arrangement. Are you getting it? Just a minute. Okay, we are using the word 9+2 arrangement. Now see, this B is attached to this A na. This B is attached to this A na. Can you see that? This something in this yellow color. Can you see it? This yellowish part. Yes, this yellowish part. Can you see that? So it is basically, this connection is basically because of the protein nexin. It is the nexin protein which is linking it. It is the nexin protein which is linking it. Can you see that? It is the nexin protein which is linking it. Nexin. What is it? Nexin. Can you see that? Nexin. Now these are the radial spokes. This part is the central sheath, and bache, here these outer and inner arms, these arms are made up of dynein protein, right? This arm is made up of dynein protein. This arm is made up of dynein protein. Which protein is there? Dynein. And bache, this dynein protein is also an active ATPase, right? Right, this dynein protein is what? This dynein protein is an ATPase. What is the meaning of ATPase? It is going to hydrolyze the ATP. It will break the ATP, it will hydrolyze the ATP, energy will be used, and that energy will help in the movement. Right, bache? That energy will help in the movement. Are you getting my point? That energy will help in the movement. Okay, bache? Manoj bache, thank you so much. I'm really grateful. Please do not spam here. Please focus here. Okay, and if you will see, again, A is complete. When you are talking about the doublet, 1A wala part is complete, having 13 protofilaments of microtubule. B is incomplete. It is C-shaped, having near about 10 protofilaments of microtubule. You can see here like this. So it is having this dynein protein. So it is an active ATPase, will hydrolyze ATP, energy will be released, which will be used in the movement. So microtubules will get the energy from this dynein protein by the hydrolysis of ATP. And when you talk about the actin, when you talk about the microfilaments, they are having myosin. Remember, myosin, we will relate it in the part, in the muscle contraction part. Myosin is that ATPase. Okay, myosin is that ATPase. So this is the difference in that 9+0 arrangement and 9+2 arrangement. Here you people can see the actual structure from NCERT. The central sheath is there. There is the central microtubule. There will be the, see, this is the central bridge also. See, A, B, and this is how they are linking. So here this interdoublet bridge is basically made up of nexin protein, and the arms are what? They are made up of dynein protein. They are made up of dynein protein. They are made up of dynein protein. Are you getting it? Yes, bache, are you getting it? This is the structure. Now I hope the it is clear to you that which will have 9+0 arrangement and which structure is having 9+2 arrangement, right? Which part is having 9+0 arrangement, which part is having 9+2 arrangement. Is that clear? Sure? Still, if there is any doubt, do let me know. Today the number of students are very less, but I am still enjoying teaching. Literally, matlab it's fun. After so long I'm teaching. No, after 2 days I'm teaching basically. But it's good. I'm feeling good. So that is why I didn't keep any class in the morning tomorrow, because tomorrow you have to revise this particular chapter. Okay, you need to revise it properly. You need to read each and every line from NCERT. You need to practice so many questions. You need to make the notes as well. Fine. So now here you can see cilia, singular is cilium. Okay, flagella, singular is flagellum. So here like outgrowths they are, bache, they are the outgrowths of cell membrane. I have explained it. Cell membrane is there, central exoneme is there, right bache, central exoneme is there. So cilia are small structure which work like oars. Obviously, you know, in the paramecium it will also help in the movement. It will also help in the movement of food towards the mouth, right, towards the mouth, right bache. So flagella are comparatively longer, responsible for cell movement, right? Just a minute bache, I don't know, just a minute. Okay, I hope. Okay, so now here you people can see, right, cilia are the small structures. Okay, this part is done. So the prokaryotic bacteria, they are also having flagella, but remember in the case of prokaryotes, the flagella, right, the flagella is made up of flagellin protein. In the case of prokaryotes, the flagella is made up of flagellin protein, but when you talk about the eukaryotes, it is made up of microtubules, right bache? It is made up of microtubules, right bache? Right? So electron microscopic study of the cilium, you know that they are covered with the plasma membrane. Core is the exoneme. It is having microtubules, right? Microtubules are running parallel to the axis, to the long axis, and the exoneme usually has 9 doublets of radially. Now focus on these words, 9 doublet, and they are radially arranged peripheral microtubules. So periphery pe 9 doublets are there. In the center we will be having what? We will be having the singlet. So central tubules are connected by bridges. They are also enclosed by central sheath, and it is connected to one of the tubules of each peripheral doublets. So this is what I have already explained, bache. So I hope the cilia, flagella and the centriole part is clear to you. Now we need to discuss the next organelle. We need to talk about the nucleus. After nucleus we will discuss the microbodies and the chapter will be over. Right? Chaitanya 24. How's Wasim sir? Chaitanya Wasim sir is doing well, but he mentioned it that no need to answer, no need to entertain Chaitanya. He's just here for spamming. Okay? So right now I'm going to ignore you. Bache, in the case of prokaryotes, the flagella, if you remember, they do not have the centriole, that is a fact, but the you know, some basic structure is there from where that flagellin will grow. They are also having, you know, that basal part is there, the hook is there, that thread-like part is there, but it will grow in a different way. Okay, they do not have that proper centriole or that microtubules and 9+0 arrangement part. Done, bache? So now the next part that we need to discuss is nucleus, a very important organelle, and again a double membrane bound organelle. Right? What is it? It is the double membrane bound organelle. Okay. It is what? Double membrane bound organelle. And when you talk about the nucleus, bache, first time, you know, it was checked by the Anton von Leeuwenhoek. Even Anton von Leeuwenhoek, Dutch microscopist. He has studied the he was working on that microscopes, no, basically. He was making that. Okay, so yeah, so first time it was observed by Anton von Leeuwenhoek, but he was not able to describe it. So first time the nucleus was described by Robert Brown in the orchid root. Remember this? It was first described by Robert Brown in the orchid root, nucleus, double membrane bound organelle. But first time, as I said, who observed it for the first time? Anton von Leeuwenhoek. Is that clear, Samiksha? Who observed it for the first time? Anton von Leeuwenhoek. Okay. Done. Anton von Leeuwenhoek. Clear? Okay. Just a minute. Done? Okay. So now when you talk about the nucleus, In the nucleus, of course, here we are having some, condensed structure. See, here also it is having the outer membrane, it is having the inner membrane, and obviously in between that intermembrane space is there. And moreover, in the case of nucleus, at points the outer and inner membrane is fused, and here you will be having some pores, right? At points this outer and inner membrane is fused, and we are going to find some pores here, right? Like if this is the nucleus, so over it there will be imagine it like a round spherical structure, so it will be having many pores, right? Nuclear pore complexes. Okay bache, so outer membrane is there, inner membrane is there, there is the intermembrane space, and when you talk about the inner membrane, it is connected here to this nuclear lamina. So this nuclear lamina is nothing, it is keeping it in proper shape, it is the cytoskeletal element, it is the intermediate filament, right? It is the cytoskeletal element, it is the intermediate filament. And here bache, in the center, here this part, it is the nucleolus. It was discovered by Fontana, yes. Fontana, the scientist's name is not important. So nucleolus. So nucleolus is considered as the ribosome factory. It is not a proper organelle, it is just a structure which is present in the nucleus. Okay, it is the ribosome factory. Samiksha, what is it? It is the ribosome factory, right? This nucleolus used to form ribosomal RNA. Leaving 5s ribosomal RNA, it used to form all the eukaryotic RNA. Okay, so nucleolus, ribosome is the protein factory, but nucleolus is the ribosome factory because it used to synthesize ribosomal RNA and proteins which are helpful for the formation of ribosome. So can I say that, right bache, can I say that when it comes to the ribosome, when it comes to the nucleus is even helping in the synthesis of ribosome? Can I say that the nucleus is even helping in the synthesis of ribosome? Yes? Of course I can. It is also the function of nucleus. Nucleus is like a central processing unit. It is going to do what? It is going to control the activity of the cell. It is having the genetic material, it is having the DNA. That DNA will form messenger RNA. That messenger RNA will form protein, right? So it is going to make everything basically. It is going to form everything. Okay, so here this nucleolus is not any organelle. It is just a dense structure present within, and it is the ribosome factory because it used to form ribosomal RNA and protein, right? Ribosomal RNA and protein. And inside we are having the nucleoplasm. You can also use the word bache, karyoplasm. You can also use the word karyoplasm. And these coverings are also known as karyo. Karyo word is associated with nucleus. So we can also use the word karyotheca for it. Right? We can also use the word karyotheca for it. Are you getting it? So nucleoplasm or karyoplasm is there, nucleolus is there. These coverings are also known as karyotheca. Right? Karyotheca. Done, bache? Okay, karyotheca. Are you getting my point? And here in between, as I said, outer and inner membrane, they will fuse. They are going to form the nuclear pore complex. So this nuclear pore complex is itself made up of 8 proteins. 8 proteins are going to make it. So from this nuclear pore complex, things will come out, things will go in. Things will come out, things will go in. Okay, things will come out, things will go in. That is how it works. Fine, that is how it works. Fine, bache? Fine. And you, if you know it, then good. If you don't know it, now you know that within this nucleus, we will be having the DNA. And in eukaryotic cell, the DNA is linear. Okay, in the eukaryotic cell, the DNA is linear. So this linear DNA will coil over the histone proteins. Remember, it will coil over the histone proteins. It is going to form the condensed structures. At the time of cell division, Mamta, it will form condensed structure. This is the chromosome. It is the chromosome. What is it? It is the chromosome. It is the chromosome. So it is a monad chromosome, right? Even it is also a chromosome, but it is a dyad chromosome. Dyad chromosome. It is having 2 arms. Fine, it is having 2 arms. So after, you know, in cell cycle you will understand it in a better way, right? In cell cycle you will understand it in a better way. So here in this chromosome, this central part is bache centromere. What is the word? Do not ever get confused in centromere, centrosome, centriole. It is the centromere. What is it? It is the centromere, and this centromere is also known as primary constriction. It is also known as primary constriction. What is it? It is also known as primary constriction. So this linear DNA, like if I talk about the humans, right? In human, the DNA is 2.2 meter long. In the humans, the DNA is 2.2 meter long. It is 2.2 meter long. So we have to adjust that 2.2 meter long DNA in that microscopic cells. So what is going to happen? The coiling will occur. So the DNA will coil over the histone proteins. It will coil over the histone proteins, and further its coiling will be done with some other proteins. So when DNA coils over the protein, then we use the word bache chromatin. Okay, then we use the word bache chromatin. Chroma word is associated with color. So it means it is going to give color with the dyes. It is going to give the color with the dyes. Dye means stain. Means with basic dyes it will provide us the color. With the basic dyes it will provide us the color. So here you can see the DNA will coil over the histone protein. Further coiling will be there. The different level of coiling is there. We will discuss it in genetics. So the structure like this will form during the cell division. Normally when you talk about the nucleus, when the cell is not dividing, the DNA will be placed like this, right? It will be tangled, okay? It will be tangled. So very highly coiled wala DNA, which is not available for the protein formation. It is going to be heterochromatin, okay? The darkly stained. And the one which is, you know, loose, it is densely packed. It is highly packed, densely packed, it is heterochromatin. The one which is loosely packed, which is available for the protein synthesis, that is, which is lightly stained, that is euchromatin. Okay, we can use this DNA for the formation of proteins. So what is the chromatin protein? What is the chroma means color. So we can use that color. Chroma means color. So basically it is giving us the color with the basic dyes. So when DNA will coil over the proteins, when its coiling has been done, then we use the word chromatin. Okay, then we use the word chromatin. So here you people can see the centromere, right? So it is the monad chromosome, which is in which the DNA content is still same. It is not doubled. This is the dyad chromosome, right? Here also this primary constriction is centromere. These are the 2 arms, right? So because these 2 arms are known as sister chromatids, right? These 2 arms are known as sister chromatids, right? Sister chromatids. What is it? Sister chromatid. So it's a DNA, it's a chromosome in which the DNA content is doubled. It is the chromosome in which the DNA content is doubled. So here you can see the centromere is covered with this basal body, and for this we use the word kinetochore, right? So actually the centromere from both the sides, it is covered with this disc shaped structure, right? It is covered with this disc shaped structure, and for that disc shaped structure we use the word kinetochore, okay? And at the time of cell division, the spindle fiber is going to attach to this kinetochore only. It is going to attach to this kinetochore only, okay? But I told you na, the euchromatin will be transcriptionally active, the heterochromatin is transcriptionally inactive. It depends upon the level of, it depends upon that packing. One is densely packed, another is loosely packed. Fine. And sometimes on some chromosomes na, at this point you will even see some secondary constrictions. Not always, but secondary constrictions. They are making the satellite DNA. Okay, so this is about the nucleus. So here you can see it's a cell organelle first described by Robert Brown in 1831. Okay, so later the material of nucleus, because it can be stained with the basic dyes, It was given the name chromatin by Fleming. So Fleming named it as chromatin. Chroma means color. Chroma means color. So interphase nucleus. What is the meaning of interphase nucleus? The cell which is resting. The cell which is just resting. It is preparing for the cell division, but it is not dividing. Right? It is just preparing for the division, but it is not dividing. So interphase, the resting phase, the energetic phase. Actually what happened now, earlier scientists thought that in interphase, because microscopes were not that good, so they didn't observe any movement in the interphase. So they were like, interphase is a resting state, but actually it is the stage when the cell is metabolically very active, when the cell is doing so many activities for the proper cell division. Okay, this is what you people need to keep in your head. So interphase nucleus. It has highly extended and elaborated nucleoprotein fibers. So what is chromatin? Nucleoprotein. Nucleic material is there, protein is there. Right, bache? So nuclear matrix is also there, and you know that spherical body nucleolus is there. Nucleolus is singular, nucleoli is plural. It is not some membrane bound structure. No, you can see that the material is condensed here. That's all. That's all. Surrounding the nucleoplasm is there, nucleopore, nuclear membrane, that's all. Okay, bache? So electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes, they are having a space, perinuclear space. Now in this nucleus, the outer membrane, it is towards its outer side, even it is also having some ribosomes. The outer membrane is also having the ribosome attached to it, and even that outer membrane is in continuation with the rough endoplasmic reticulum. Remember, bache? It is even in continuation with the rough endoplasmic reticulum. Okay, the outer membrane. The outer membrane. Okay? Okay? So, the outer membrane usually remains continuous with endoplasmic reticulum. Okay? And at the number of places, nuclear envelope is interrupted by pores, and I told you that pores are formed by the fusion of outer and inner membranes, right? So with the help of these pores, things can move out and in. RNA protein can move out. Okay, bache? So both the directional movement is possible. Okay, bache? That is not required for you, Srinu. Not required for you. Done? So there is only one nucleus per cell, you know that, but some cells are binucleated as well. Some cells are binucleated as well. Remember, bache, paramecium. Paramecium is having macro and micro nucleus. Paramecium is having macro and micro, and moreover, in sieve tube cells, in RBCs, at the stage of maturity, they are enucleated. Okay, when they are mature, they do not even have the nucleus, they are enucleated. And moreover, if a cell is having so many nuclei, then what is the word that we use? We use the word senocytic condition, that is multinucleated condition. Multinucleated condition, and for that we also use the word syncytium. Syncytium, multinucleated condition. But sieve tube cells are the part of phloem. Okay, okay, right? So actually there is a difference in the word senocytic and syncytium. When the word is syn means fused. See, when there are so many cells together and they are having common cell membranes, they are having common membranes. When these common membranes fuse, and then you get this multinucleated condition, so at that time you use the word syncytium. Okay, at that time you use the word syncytium. Okay, so ultimately the meaning of syncytium and senocytic is multinucleated condition. It is the multinucleated condition, that's all. Okay, so see, some mature cells, even they lack nucleus. I told you erythrocytes, sieve tube cells of vascular plant. Okay, so nuclear matrix is nucleoplasm. I have explained it. Okay, so in the nucleolus, active ribosomal RNA synthesis is there. Okay, bache. So I hope it is clear to you. If still there is any doubt, do let me know. If still there is any doubt, do let me know. Just 10 minutes more and the chapter will be over. So here you can see the central centromere. These two arms covered together by disc-shaped proteins structure, kinetochore. So it is the kinetochore to which the spindle apparatus will be attached, right, to which the spindle apparatus will be attached. Done, bache? So as I said, chromatin is having DNA, histone proteins are there. So histone proteins are positively charged, they are basic proteins. Okay, they are positively charged, they are basic proteins. You can mention it in your notes. Even if you are preparing for NEET 2025, you should mention it in your notes. Okay, so in histone proteins, you are having glycine and arginine. Lysine and arginine. Okay, lysine and arginine. So some non-histone proteins and some RNA is there. That's all, bache. So each chromosome is having primary construction. I have already explained it. So on the basis of position of centromere, you are having different type of chromosome. Like if two arms are equal, then it is going to be metacentric. Okay, on the basis of this position, you can see this. See, two arms are equal, right? Metacentric. Then submetacentric, right? Then acrocentric and telocentric. This is important. Okay, so acrocentric when centromere is situated close to its end, forming one extremely short arm and long arm, and telomere when the chromosome is having a terminal centriole. So this condition is not there in the case of human cells. You are not going to find it here in the human cells. Okay, okay, so if there is any doubt, do let me know. So if up to this part all clear, please let me know in the chat section. Quick, bache. Quick. Done? Yes? Sure? So on the basis of the position of the centromere, right, we are having, so metacentric when arms are equal, submetacentric, you know, when it is not in the centromere, centromere is not there in the center, it is towards one end, but the difference is not that much, you can see here. Okay, then there will be the acrocentric and finally there will be the telocentric. So telocentric mein you people can see, right, see in telocentric, in case of acrocentric, the centromere is situated close to its end, forming one extremely short and one very long arm, whereas in telocentric, chromosome has a terminal centromere. In humans, we are not going to find it. Okay, in the humans, this case will not be there. Now, one more homework for you. You people will tell me, okay, you guys are going to tell me that in the case of humans, we have 23 pairs of chromosome. So you have to tell me which chromosome is metacentric, which is submetacentric, which is telocentric, which is acrocentric. Telocentric is not there, but this is what you need to do. Okay, this is the first homework. Secondly, this satellite and secondary construction is there, na bache? Secondary construction is the one we cannot color it, right? We cannot stain it. It will not get stained, this secondary construction, right? And this secondary construction is also known as NOR, nuclear organizer region. Right, this is known as NOR, and here to the end of this secondary construction you will find this satellites. You're going to find these satellites, and even when you talk about the DNA, we discussed the satellite DNA in your class 12th. So when we talk about the satellite DNA, we consider them as some, you know, some repeated sequences. Some DNA is actually made up of nitrogenous bases, right? So some repeated sequences are present there, and they are guanine rich basically. You can say that they are guanine rich sequences here. So even in class 12th you will discuss about mini satellites, macro satellites. So they will somewhere help in differentiating two individuals. They will somewhere help in DNA fingerprinting. We discuss variable number tandem repeats. That is a very different concept. So right now I'm not going to discuss that, but as of now I'm just telling you about the satellite DNA. Guanine rich it is, right? And secondary construction, it is something which will not get stained. It is also known as NOR bache, right? So that is what you guys can mark. Fine. and I already told you about the euchromatin and the heterochromatin. So still, if there is any doubt, please let me know. Do you have any doubt from this part? So rest you have to do the homework. And non-staining secondary construction at a constant, this give the appearance of a called the satellite. Okay, done. Now, but some small organelles are also there. We used to call it microbodies. When you talk about the microbodies, you have two examples: peroxisomes, also known as uricosomes, and one is glyoxysomes. Right, we have two examples: glycosomes and peroxisomes. These are the two examples that you need to know: glyoxysomes and peroxisomes. Glyoxysomes and peroxisome, and this is what you people need to remember. Okay, bache, when you talk about the peroxisome, they are present in both, right? They are present in both animal cell and plant cell. They are present in both animal cell and plant cell, but when you talk about glyoxysome, they are present only in plant cell. Okay, and you are going to find them in the case of in the seeds, where fatty acid metabolism is going on. In the germinating seeds, you will find too many glyoxysomes. In the germinating seeds, you will find too many glyoxysomes. And these peroxisomes, they are even present in the cells which are undergoing beta, where there is the beta oxidation of fatty acid, right? So it is playing role, this organelle is playing role in the beta oxidation of fatty acids, and this is what you people need to remember. Okay, so they peroxisomes, they are involved in beta oxidation of fatty acids, right bache? So in plants and animals you are going to find it. Okay, you are going to find it. And moreover, in the peroxisome there is one enzyme catalase. Okay, so it is going to it will help in that H2O2 metabolism. H2O2 is your water basically it is your peroxidase, isn't it? Peroxidase. So catalase will act on it, it will break it into water and oxygen will be liberated. Okay, so catalase enzyme is there which is going to act on that peroxidase. I'm mentioning, peroxidase is an enzyme, it is peroxide, H2O2. Water peroxide, it is H2O2. So catalase will act on it and it is going to break it. So peroxisome is having this enzyme catalase. This is important. Okay, this is important. Done, bache? Okay, so the role of peroxisome is important. So the cells having beta oxidation of fatty acids, in that also peroxisome is playing the role. It is having the enzyme catalase which will act on water peroxide. It will break it into water and oxygen, of course. And moreover, peroxisome, this organelle is even responsible is in photorespiration. In the case of plants, we talk about a wasteful process na. In the case of plants, we discuss about a wasteful process. We used to call it as photorespiration. Even we used to call it as C2 cycle. So in that C2 cycle, in that photorespiration, this peroxisome used to play the role. Okay, it used to play the role. Okay, bache. And when it comes to the glyoxysomes, as I said, it is present only in the plant cell. In the germinating seeds, you will find it, right? And moreover, bache, in the glyoxysome, they are playing role in glyoxylate cycle. That's all. This is what you need to remember for this microbodies. Okay, so it is playing role in what? Glyoxylate acid cycle. Done, bache? Done. So in fatty seeds, the glyoxysome will be present. So ultimately, glyoxysome will convert fats into carbohydrates. So this is all about the microbodies. Done, bache? This is all about the microbodies. So this is what you need to remember. So finally we are done with the chapter. I'm very happy. Okay, so we have covered everything properly. Everything properly we have covered. Still you guys are having any doubt, feel free to ask your doubts in the comment section. I will definitely answer it. Do revise the things properly, right? Be the part of these classes regularly. It is on YouTube. See, let me tell you, on YT we are taking the Achievers batch. Okay, many students are confused, so I'm telling you. So obviously it is free of cost. Here in this batch we will cover high weightage chapters. Okay, we are going to cover high weightage chapter, but on Unacademy platform we are taking Avengers batch, and it's a paid batch where your all syllabus will be covered. We will finish the 11th syllabus first and then 12th. in 6 months we are going to complete your course with question practice and everything, right? This is what we are going to do. Remember this thing. So bache, we are going to start the classes for Avengers batch on 25th of May. Right, you will get your first class on 25th of May, and now because of Unlock 20, right, you are getting this batch in just 9599 rupees. So till 26th of May, right, if you want to enroll in this batch, you should enroll very quickly because we have limited number of seats, first thing. Secondly, right, from 25th of May we are going to start the classes. If you will think that I will join later or I will join after the result of NEET examination, obviously you people will end up in creating the backlogs. So avoid that backlogs, be the part of this batch, because till 26th of May, right, 12 AM of 26th of May, you will get this batch in just 9599 rupees. But after that, the course of the batch will be 14999. Okay. So how can you join the batch? You just need to go to any of our video, click on the batch link, add your number, add your email. See, Avenger coupon code is applied, but because of the special offer which is valid till 26th of May, you will get extra discount of 20% and the batch will be there in 9599 rupees. So do it quickly, be the part of this batch quickly, because here you will get that personal connect, right? Your personal doubt solving will be there, the question practice will be there, right? So you are going to get everything that you need for cracking the NEET examination. So do hit the like button, do mention your homework in the comment section. If there is any doubt, even you can mention that doubt in the comment section. So thank you so much everyone. We are done with the chapter. So day after tomorrow we will be starting cell cycle and cell division, which is very important to understand the basics of human reproduction. So attend that session properly. Okay. So thank you so much. Good night. Tada. Bye bye.